Hello everyone, today I'm gonna talk about photosynthesis. First, let's start with some definitions that you should be aware of in this chapter. I recommend you to pause the video and write down these two definitions. Also, you should recall your knowledge for the equation of photosynthesis. Basically, two sets of reactions are involved in photosynthesis. Let's discuss about the light-dependent reaction first. The light-dependent reactions include the splitting of water to get hydrogen ions, which in other words called photolysis, and synthesis of ATP, which is photophosphorylation. Light-dependent reaction occurs in thylakoid of grana, where first stage of photosynthesis happens. It only takes place in the presence of suitable pigments that absorb certain wavelengths of light. In addition, this light energy is needed for photolysis of water and synthesis of chemical energy, which is ATP. In photophosphorylation, it can be both cyclic and non-cyclic. Cyclic photophosphorylation only involves photosystem 1. When the light energy passes to primary pigments through photosystem 1, the excited electrons are emitted from chlorophyll molecule, and this is called photoactivation. Then the excited electrons are captured by an electron acceptor and passes back to a chlorophyll molecule via chain of electron carrier. ATP is made here and passes to the light-independent reaction. Next, non-cyclic phosphorylation involves both photosystem 1 and 2. Light is observed by photosystems and excited electrons are emitted from primary pigments of both reaction center. Then, these electrons are observed by electron acceptor and pass along chains of electron carrier. Thus, photosystems are positively charged now. Primary pigments receive the electrons again by undergoing the photolysis of water. This is the G-scheme of electron flow in photophosphorylation. Cyclic photophosphorylation only happens here, which is photosystem 1, whereas non-cyclic photophosphorylation happens at both photosystems. To talk more about the photolysis of water, it is basically splitting of water molecule into hydrogen, electrons, and oxygen. Photolysis of water only happens in non-cyclic photophosphorylation since the enzyme that breaks down water molecules is secreted in photosystem 2. This is the equation for photolysis of water. In this reaction, oxygen acts as an waste material. Hydrogen ions can combine with electron from photosystem 1 and carrier molecule NADP to give reduced NADP. To the reference to Robert Hill, who claimed that isolated chloroplasts had reducing power and liberated oxygen from water in the presence of an oxidizing agent, we can conduct this knowledge to investigate the effect of light intensity or of light wavelengths on the rate of photosynthesis of a suspension of chloroplast. Blue-dyed DCPIP can be used in this experiment. When it is reduced, the color changes blue to colorless. Next reaction is called light-independent reaction. You should memorize this cycle because it's very important in this chapter. This reaction takes place in stroma and TP are mainly used to regenerate RUBP and remainders are condensed to hexose phosphates for producing starch, cellulose, and glycerol and fatty acid. Also, the enzyme Rubisco catalyzes the reaction when carbon dioxide and RUBP combines. Three factors affect to the rate of photosynthesis. First one is light intensity, second is temperature, and the last one is CO2 concentration. However, although the rate of photosynthesis changes, it eventually reaches maximum point where the graph becomes constant. In the condition of high light intensity and high temperature, the rate of photosynthesis increases. However, in the condition of low light intensity and high temperature, the rate of reaction does not indicate significant change. We cannot say that temperature does not affect the rate of photosynthesis at all because the rate of photosynthesis changed by temperature in other researches when the variable was only the temperature. For the limiting factors, low concentration of CO2 becomes rate limiting factor, whereas high concentration of CO2 makes other conditions to be a rate limiting factor. C4 plants are tropical plants which get excess sunlight. Photorespiration happens inside C4 plants. RUBP combines with oxygen, which is waste material rather than CO2. Thus, it reduces the efficiency of photosynthesis, and for avoiding this, C4 plants have a special vascular bundle arrangement to prevent oxygen to penetrate into the mesophyll cell. You should also remember this special cycle for C4 plants. To briefly talk about photosynthetic pigment before we proceed to next, primary pigment contains chlorophyll A and B. Chlorophyll A indicates yellow-green color and chlorophyll B indicates blue-green color. Since the primary pigments observe red and blue-violet region, they both indicate green color. In addition, accessory pigments contains carotenoid, which is B-carotene, and xanthophylls. B-carotene indicates orange color and xanthophylls indicate yellow color. Accessory pigments mainly observe blue-violet region. Let's talk about the absorption spectrum first. This graph is for the observance of different wavelengths of light by pigments. As you can see in this graph, chlorophyll A and B observe blue and red light, but carotenoid observe violet and blue-green light. For the action spectrum, it is for the rate of photosynthesis at different wavelengths of light. It also shows the effectiveness of different wavelengths. Shorter the wavelength, greater the energy it contains. In order to extract the chlorophyll, we should do paper chromatography experiment. This equation is for calculate the RF value. Carotenoid has highest RF value and chlorophyll B has lowest value. The value of chlorophyll A is located between the value of carotenoid and chlorophyll B.